Okay, so if we look at the biological molecules, we have carbohydrates, lipids, and proteins. And we need to know the structure of all three as well as their bonds. So if we start with carbohydrates, they always have this general formula of CnH2OY because oxygen is always in the ratio with hydrogen of 2 to 1. That's why they're called carbohydrates. Carbo from carbon, hydrates because of this ratio. So if I have 6 carbon molecules, okay, which is our general monosaccharide that we always use, it means that it's going to be H12O6 because those are always in the ratio of 2 to 1. So our simplest form of a carbohydrate is called a monosaccharide. Then when you join two of those together, you get a disaccharide. And then a whole string of them together is a polysaccharide. So anytime you see the word with saccharide, it refers to sugar, and there we're referring to our carbohydrates. So our most common carbohydrate is glucose. So now this is the straight line structure of glucose. So we've got C, that's double bonded to an O. On the second one, there's an OH group, that's to the right, then an OH to the left, and then to the right, all the way down. So there's 6 C's, 12 H's, and 6 O's. So now if we look at that actual structure between them through molecules, I can start with carbon 1. So on carbon 1, there is a double bonded O on that one side. And then if I move to carbon 2, the OH goes to the right. And then carbon 3, it's on the left. 4, it's on the right. 5, it's on the right. And 6 is on the right. So when that happens, and it actually is a straight line structure, you can see that it's not in an actual straight line because it's all curved. And when that happens, this carbon OH group on carbon 5 becomes close enough to carbon 1 with that double bond. So when there's close proximity like that, they can break and they can bond together. And that's when we can form a ring shape like that. So you've got a glucose ring with oxygen in it as a 6-sided ring. And then the OHs are facing certain ways. So if we look between our carbon 5, there, and there, you end up with this carbon ring shape. With the 6th carbon on the outside and an oxygen in the middle there. So if we look at that, we've got carbon 1, 2, 3, 4, 5, 6. Okay, and now if you look at the arrangements of those in that ring, so there's my carbon ring. So there's the carbon 1, 2, 3, 4, 5, and there's the oxygen, and there's carbon 6 at the top of the ring. So now there is an arrangement in space of these OHs, because you see this one is facing downwards, and then this one goes up, and then that one goes down. So that arrangement there on carbon 2, 3, and 4 is always like that. It's always down, up, down. So when we're drawing that, we can draw them because now it's going to go down, up, and down on those ones. On carbon 1 over there, this one is facing downwards. But that depends on which of these double bonds got broken, because we could have broken either one of them. So now this one happens to go down, but we could have broken the other one, which would go up. and that would create two different kinds of glucose. So either it's going to go down, or I would have another ring. Where the OH on carbon 1 would go up. And then 2, 3, and 4 are still down, up, down. So now that is still carbon 1, 2, 3, 4, 5, and 6. But now the OH there is facing downwards and there is facing up. So now when it faces downwards, we call it alpha glucose. And you can remember it because when you draw the sign for alpha, you go downwards because it's alpha glucose. So by going down, that you can remember the OH faces down in alpha glucose. So we call that one alpha glucose. When we draw the sign for going up on that one, we can say beta glucose. And beta you can draw by going up and around. So when you draw that sign for beta, you go upwards. So the OH facing up is because it's beta glucose. So we have alpha glucose going down and beta glucose going up. And that's how we name them. So they're two different molecules because that carbon 1 is either going down for alpha or up for beta. And that's then the ring structure of glucose. Now we can draw that ring structure without the carbons in there. So we can just show the oxygen, and then you show each of the carbons as a point, because each one of those is carbons, and then you can fill in the OHs. And our last carbon that's outside of the ring. Doesn't matter which way you draw it, it's still the same structure. This one just shows no carbons, this one shows the carbons. But this is still alpha because that goes down, that goes up for beta. So then the other structure that we could have is of fructose. So this is glucose, which is C6H12O6. But then another C6H12O6 molecule is fructose. So they are isomers because they have the same formula, but they have different structures. So we have glucose, fructose, and the other structure that we also have is galactose. And those are all our monosaccharides that are most common. So now the difference between fructose is that it's still got 6 carbons, 6 oxygens, and 12 hydrogens, but as we can see the difference between there and there, that double bonded O is on a different numbered carbon. It's on the second one. But then the rest of the molecules are all the same. So just by switching those around though, and moving that position, it changes the ring structure because now a ring is going to form between that and that, which means 2 carbons are now outside of the chain because that's now carbon 1. And on carbon 2, it joins with 5. So now you get this 5-sided ring. And now with that 5-sided ring, the OH on carbon 3 and 4 still goes up and then down. It's just now carbon 2 where it changes. So that is when we have the same formulas, but they make a different structure. So this makes a different molecule. So we just have to keep in mind that if we see this, it's going to be fructose, whereas these are glucose. So now the special thing about these carbohydrates and these monosaccharides is once they've made a ring structure, they can join together. So here I have a glucose ring. On carbon 1, the OH is facing downwards. 2, 3, 4, 5, 6. So since this is facing downwards, that means this is alpha. Then on this one, it's carbon 1, 2, 3, 4, 5, 6. Also alpha because of that. But now if those rings are joined together, so if we have a ring and we line it up with another ring, sometimes the OHs line up in the same way. And because they are in the same orientation, like here, they're both facing downwards, they can line up together close enough that those can join together, and that can come off because there's an H and an OH can come off as H2O, so as water. So now, with that comes off as water, it means this ring joins to that ring with an O in the middle. So now, we're going to have a ring next to another ring. The oxygens are still in the ring over there. We still have the OHs on 2, 3, 4, going down, up, down. And carbon 6 is still outside the ring. And then the OH on carbons 1, 2, and 3. And 6 still outside the ring. But now between 1 and 4 that we had over there. So that's carbon 1 and that's carbon 4. I took the H2O away and I joined them together with that oxygen in the middle. So this bond that gets made is called a glycosidic bond. So in carbohydrates, you join the rings together with an oxygen bond like that, and it's called a glycosidic bond. So now we have a glycosidic bond between carbons 1 and 4, and these were alpha molecules. So we can say it's an alpha 1, 4 glycosidic bond. So that's the full name of that bond. Plus, I made a water molecule. So now when I join things together and I make water, so by adding that, because I've joined them together and I've made it, the type of reaction is a condensation reaction. So my condensation reaction is when I make water and I join things together. If I want to go backwards and I want to split this molecule up by adding the water and breaking it and going back to the original, I call that hydrolysis. Hydro means water, lysis means split. So hydrolysis is to split them back, and to make them is condensation. So if I want to go that way, it's condensation. If I want to go that way, it's hydrolysis. So now that's our most common form of bond for our alpha 1,6 glycosidic bonds. But now, remember, I also have a beta molecule. Now, a beta glucose molecule is when the OH on carbon 1 faced upwards. So here, because I've got carbon 1, 2, 3, 4, 5, 6, on carbon 1, that's going up. So that is a beta molecule. And over here, 1, 2, 3, 4, 5, 6. On carbon 1, it's going up. So there's a beta molecule. So now with these beta glucose molecules, on carbon 1, because it's going up, when I line it up next to the other one with carbon 4, those are in the wrong direction because they're not going to line up anymore when they're going across. So in order to bond these together, I have to flip one of the molecules. So I take this molecule and I flip it 180 degrees. So if I take this molecule like that, I flip it over so that everything that was facing up is now facing down. So there was carbon 6, now carbon 6 is over there. So I have 1, 2, 3, 4, 5, 6, because I took this one and I flipped it over. So now everything that was at the bottom is now at the top. So now in carbon 1, because it's upside down, where it was up, it's now down. And then where it was down, now it's up. Where it was up, now it's down. And where it was down, now it's up. Okay, so now I flip that around. And if I draw this other molecule that I have again, carbon 1, 2, 3, 4, 5, 6. So now this is the same molecule that I redrew. Now I can see that the OH that was facing up now lines up with that other OH. So now those two molecules are in the same direction, in the same place. So that can come off as water and they can join together. And if we join them together, I now have my one molecule that's facing upwards, that's regular. And then I have my other molecule that's facing the opposite way because I rotated one by 180 degrees. So now I've got down, up, down. And here I've got so I flipped them. I flipped this one and this one's still right. But now since that was facing up, my glycosidic bond that I'm going to make in the middle is now going up. So this is still a glycosidic bond. I still made H2O. It's condensation. It's just that now where this one was facing downwards, this one is now facing upwards. But now if I bring another beta glucose molecule in there, this one is now facing the right way because I flipped it. So you only need to flip every second molecule in the string. So every second molecule in a beta glucose is rotated 180 degrees so that they can line up and make glycosidic bonds. And where this one is now facing upwards because I used beta molecules, I call this a beta 1,4 glycosidic bond. The other one was alpha 1,4 and now this one is beta 1,4, but it's still got the same number. Okay, so if I look at my names of all my carbohydrates, so I have my monosaccharides, glucose, fructose, and galactose. So those are all my single rings. When I join my rings together, I make disaccharides. So 2 glucose rings together makes maltose. A glucose and a fructose ring makes sucrose. And a glucose and a galactose ring makes lactose. Then polysaccharides is multiple rings. So I have starch, which is the storage form of glucose in plants, which is made up of long strands of molecules with alpha 1,4 and alpha 1,6 glycosidic bonds, which just means that in a molecule like this, since there's 1 and 4, I can also join carbon 1 to carbon 6, and I can get chains. So what will happen in a starch molecule is I can have a straight chain of rings, and then I can join at carbon 6 to get a new chain, and carbon 6, and that's how we get a molecule that is called branched. So with alpha 1,4 and alpha 1,6 bonds, you get these branched chains. And starch is made up of amylose, which is alpha 1,4 bonds and a straight line, and amylopectin, which is alpha 1,4 and alpha 1,6, which is branched. Glycogen is the storage form of glucose in animals, and it is just one big branched molecule of alpha 1,4 and alpha 1,6 glycosidic bonds. And then cellulose is made up of beta 1,4 glycosidic bonds. So remember, in a beta molecule, it's made with beta glucose, and every second ring is rotated 180 degrees. And cellulose is there for structure because it's the structural cell wall in plants. So because of those beta molecules, it makes cellulose really, really strong and for structure.